Monash University in Australia, and he won several research awards for his PhD work, including Vice Chancellor's Commendation for Doctoral Thesis Excellence and the Best Engineering PhD thesis. After completing his PhD, Dr. Khan worked as a postdoctoral researcher at McGill University in Canada, and his work was on biosurface engineering and biomolecular simulations. Since 2013, Dr. Khan has been working as a full-time faculty member at the Chemical Engineering Department of Bangladesh University of Engineering and Technology. In 2015, he worked as a visiting professor at McGill University, and from 2016 to 2017, he worked as a visiting scholar at Texas A&M University. He also served as a technical advisory group member of the World Health Organization from 2020 to 2021, and he was the Chief Sustainability Officer, or CSO, of the Vaccine Co-Group in Bangladesh from 2021 to 2023. And he has also received major excellence awards. He has been authored or co-authored of more than 100 published articles and manuscripts. And Dr. Khan, we are so honored to have you today with us. Thank you so much for joining us. And the stage is yours. Thank you.
Thank you very much. And thank you for introducing me to the audience. I didn't realize that it's that long introduction.
I think we can deactivate my microphone.
So, can you see my screen? Hello?
Hello. We have your screen.
Okay, wonderful. Thank you very much for having me today. And it's a pleasure to give a presentation in front of you. And it will be more as a holistic presentation, just like the directions where we're working on. So feel free to stop me if you want to know more detail. And like you're always welcome to ask me any relevant questions. So you can ask me after the presentation or during the presentation, so don't mind. Okay, as you already know by the previous introduction, that I got opportunity to work in many places, including academia, NGOs, and industries. So I got to work with different universities like Monash University, McGill, and Texas A&M University, and also with WHO, United Nations, USAID, Europe. It's a mixed kind of experience. Each sector has its own advantage and limitations. So we'll discuss more and how it has created new opportunities in research. That also we'll discuss. And from this presentation, my expectation would be to open a new window of opportunity to have joint collaboration research program with you. Maybe at some point we'll invite you to come to visit Bangladesh, and our students can also visit your place. So as mentioned, I completed my undergrad from chemical engineering back in 2004 from Bangladesh University of Engineering and Technology. It is one of the oldest engineering universities in Southeast Asia. And our chemical engineering department is perhaps the oldest in Indian subcontinent. And after I graduated, in my undergrad, I got opportunity to work on industrial waste management. So after I graduated, I joined as a project engineer in one of the pollution abatement project run by Stockholm Environment Institute, University of Leeds, UK, University of York, and Bangladesh Center for Advanced Studies. So it was kind of very multi-disciplinary and multi-dimensional, and that got me opportunity to work directly with industries and also start working on publications. So through those projects, we published a few important publications there, and after that, I moved to Australia to do my PhD from Monash University. From publication point of view, it was quite a successful PhD program, and also it secured different kinds of awards. But during my PhD, I started working as a sessional teacher in Australia. They don't call it TA; they call it sessional teacher. So I used to teach transport phenomena, chemistry and chemical thermodynamics, reaction engineering, bionanoengineering, fluid mechanics, so that's that sort of courses. And after completing my PhD in biotechnology, it was more on the biosurface and biotechnology discipline. So I moved to McGill, Canada, that is in Montreal, to continue my research on biosurface engineering and biomolecular simulation. And if you know, McGill is quite strong in fundamental research. And we all know about Rutherford atomic model. So Rutherford was a professor of McGill, and McGill won a good number of Nobel Prizes over there. And during my postdoctoral research, I started working on different grant writing. Not every writing was successful, but I got to know how to write for grants, and that helped me in my later stage. And one very interesting thing happened while I was doing my research in McGill. I stayed there for 3 years. In my research group, I had good friends, and few of the friends, in fact, a good part of the friends, they came from Sharif University of Technology, and we got to know about the MIT of Iran. So we know about Sharif University long before we officially get to know each other. And if you see, that is like in my research group, I was a postdoc and they were doing their PhD. So I invited them at my residence. You see Dr. Zainab, she is now well established. She is working as an associate professor of McMaster University. Dr. Leila, she is working as a global project engineering at BioNTech. It is in Germany. Dr. Amir Sheikh, a very nice fellow. He is working as an associate professor in Penn State University. And then Dr. Salman, he is the president of Candle Lithium. I think it is his own company in British Columbia. So my all my friends from Sharif University, they live in Canada or Europe. So I'm the only person who came back for good to build up my career in my native country. So, and I really congratulate Sharif University for generating very high talented researchers who are just contributing science and technology over the years. So congratulations and thank you. So in 2013, I came back to Bangladesh as an assistant professor. I started working. Okay, it's kind of interesting. So even when I was talking to Dr. Haniye, we discussed that. Like in Bangladesh, it's very difficult to get any grant. And my research experience, mainly from my PhD and postdoc, was biotechnology and very sophisticated biotechnology. Okay, so one senior fellow from my university, he suggested me to have one glamour research program and one research program which would be very relevant to the country, and one research program which will earn me different kind of grants. So that's why I build up one research group that is called BET Research Group, BET, Bio and Environmental Technology. And we keep the biotechnology part as the glamour one, and the environmental part as the bread and butter, because we earn good amount of research grants from that. In total I earned something around $1.5 million. I'll discuss later on. And during COVID time, I started working for WHO in their core team to set standards for biomedical devices and PPE. And there I got to learn about different aspects of PPE. And in from 2023, I keep working for the department as a full-time professor. In the meantime, we secured different research grants from WHO and European Research Fund, and also for bio incubator. So in total, my personal research fund is about $1.5 million in last few years, and as a part of the department, we won another $1 million. So it was about $2.5 million in total. Okay, and since I already had some experience on PPE, and also I had some interest to work with industries from the very beginning. If you remember, once I completed my undergrad, I joined in industry-related research. So I got one offer as an executive director of Beximco Health and PPE. Now, Beximco is one of the largest business conglomerate in Bangladesh. They have very strong footprint in textile, pharmaceutical, ceramics, media, television, IT, energy, banking, PPE, fashion, technology, solar. So it was a good opportunity to work with them from the top-down approach. They also have 70,000 workers. So I took that opportunity. I took a full leave from BUET to join there as an executive director. And later on, Beximco assigned me as their chief sustainability officer. So as a part of CSO, I used to work with the top management, HR, supply chain, production unit, engineering, utility and maintenance, power generation, chemical storage, effluent management, market and merchandising. From 2023 onward, I keep working with industries as an honorary director and advisor. So it's not only about Beximco, I'm also associated with few other industries, local industries. And that gave me opportunity to get first-hand industrial data. That also I will explain. So my teaching and research philosophy, since I have experience in academia, industry, and development sector, I like to combine them together and provide problem-based teaching in the classroom. Also, I try to train students to solve open-ended engineering and scientific problems, to have industry academia collaboration. In recent master's and PhD thesis, I'm offering all the projects under industry academia collaboration. And that gave me cash and kind opportunity to work with industries, because in most of the industries, they also have their accredited lab. So I can use their lab also. And last but not the least, I aim to produce industry-ready graduates. So it's a very interesting. I'm not sure about the condition in Iran, but in Bangladesh, we teach our students with all the advanced mathematical and engineering problems. But often we find when they go to the industry, then they're not truly industry ready, because industry as a fresh graduates, they don't expect much. They expect the student should have very strong communication skill, understanding about the technical terms. They don't expect the student will just design one reactor from the very first day. So my aim is to produce industry ready graduate, and I'm offering a series of courses in graduate level like technology convergence and new product development, biomaterials, food processing and preservation, fermentation technology, biochemical engineering. And in undergrad lab, I mainly teach third year and fourth year courses that includes economic and management of chemical process, food processing, preservation, fertilizer, pulp and paper, biochemical engineering, industrial pollution control. And also I teach one preliminary course, that is engineering thermodynamic 1, so that I can have some early interaction with fresh students. And in research, I prefer to work on biotechnology and industrial sustainability. Under biotechnology, I work on point-of-care diagnostics. There's some work on molecular simulation, biofuels, and food safety. And under industrial sustainability, I work on sustainable raw materials and a few other items I'll explain later on. So let's focus on the current research or the ongoing research on bioactive papers, the already completed projects and ongoing projects. I'll go through all. So now, what is bioactive paper? Different research groups, they define bioactive paper in different way. Like Professor Bob Pilton from Canada, he said that a paper that will detect, capture, and deactivate water and airborne pathogens could be called bioactive paper. Professor Jill Garnier from Australia, he says that papers optimizing the biomolecular activities on a surface in terms of biofunctionality and biocompatibility. And Finland VTT Research Group, they define bioactive paper as the paper-like products, cardboard, fabrics, and their combinations with active recognition and or functional material capabilities. So, though they define bioactive papers in different ways, but all research groups target to develop low-cost bioassets for health and environmental purposes. Sorry, I just have some flu because of some allergic reaction. If you find it difficult to understand what I'm saying, please feel free to stop me. I'll slow down and I'll try to re-explain. Okay, now, though the bioactive paper research is getting momentum in recent years, however, the concept of bioactive paper is quite old. It's more than 200 years old. The first paper test strips to detect sugar in urine was introduced back in 1850s. In 1880s, a very famous scientist, Gay-Lussac, introduced pH paper, the pH paper that we use, that was introduced back in 1880s. In 1880s, also the urinary test papers brought to the market. However, very small scale. Now, in the Second World War, uh, uh, some very good investment was done to explore bioactive paper commercialization, and one specific item named Isel bioactive paper toilet paper was introduced because during the Second World War, lots of hygiene related issues were going on, and people are getting diseased and infection, so they needed to use antibacterial toilet paper. And this is one of the commercial ad from the Second World War. If you see, it's right, it's written that each smooth sheet is medicated with Isel germicide. Now, in 1950s, urinary test papers commercially produced, and in 1970s, pregnancy test strips were brought to the market to detect HCG hormone in urine. So, though it was introduced back in 1850s, but it took more than 100 years to get commercial recognition. And in recent years, we get lots of antibacterial hand wipes and paper towels. Now, few very important breakthrough done by Professor George Whiteside from Harvard University. He works on biophysics, he and his team, and they develop 2 or 3-dimensional paper-based microfluidic devices, synthesis of DNA on paper for gene detection, paper-based microfluidic electrochemical sensing. And for the first time, they introduced a single paper-based device which can detect glucose and protein at the same time. And then Professor Paul Yeager from University of Washington, he has many patents under his belt. So he developed low-cost diagnostics for enteric pathogens and emerging diseases like diarrhea and dengue fever. And Professor Jill Garnier and his team, so I was a part of that team. So he worked on point-of-care diagnostic system aiming for antibody enzymes and biomolecular printing. So the main critical questions about having bioactive papers as paper filters and diagnostics. First, does biomolecule immobilization affect reactivity and shelf life? Like once we put the biomolecules on paper, does it stay alive or does it stay functional? We need to understand. What is the temperature effect? Like I just prepared one diagnostic device and I send it to a desert area where the temperature is 45 degrees Celsius at 50 or 60% relative humidity. Will it still be functional? Or if I send it to a country like Bangladesh where the temperature is 36 degrees Celsius and humidity is 85 plus, will it be functional again. Then polymer effect on retention and biomolecular activity. So to keep biomolecules on paper, sometimes we use polymer, whether the polymer interacts with the biomolecular activities or not. Then can biomolecules be inkjet printing? Like we can put biomolecules on paper and we can develop diagnostic devices, but to go for commercialization and bulk volume, we need to get them printed. So can we print them? And can biomolecular interactions be modeled to help experimentation? So can we do simulation to understand experiments before we even enter into the experiment? So let's get all the answers one by one. First, inkjet printing of biomolecules and paper microfluidic channels. So we used traditional inkjet printer. You know, inkjet printer can be bubble jet or piezo. So in bubble jet, it is the thermal physical activity, and in piezo, it's the kind of the electronic signal. But for better resolution, we always go for thermal. So if it is a thermal bubble jet printer or inkjet printer, does it destroy all the biomolecules through the printing? So that was the first question. So we printed different enzymes. If you see, we printed horseradish peroxidase, that is an enzyme, on paper, and we can see that the printed enzyme is giving positive signal. So that means printing doesn't destroy enzymes on paper. And also we printed protein solution on scaffold. The protein was named albumin FITC. So we can still see some signal from albumin FITC printed scaffold. And to develop hydrophobic microfluidic channel on paper, we used AKD solution. I will show how it works. So what we did, we printed different hydrophobic channels on paper, and then we printed different enzymes or biomolecules inside of those channels. Let's see whether it still works or not. So those are the printed channels, and we place different enzymes, and then we use substrate to react with enzymes. So that was the simulation, and that is the real life printing. So we can see that we can develop different kind of patterns on paper using AKD solutions, and then print precisely biomolecules inside of the channels. And there is one video demonstration. So we printed, you cannot see maybe, but sorry, now we can see. So we're placing substrate at the middle of the paper diagnostic or paper channels, and it's wicking through the microfluidic channels and make all the reactions. So you can do lots of very precise printing through bubble jet or thermal inkjet. So normally a bubble jet can generate a droplet of picoliter size. On the other hand, piezoelectric produces the droplet much larger than the bubble jet. So the resolution wise, bubble jet is more convenient if you want to draw parallel lines using different proteins. And we secured the whole concept by applying for patents. So it is already patented. The technology, you see US patent. We also have Chinese patent, European patent, and a few other countries. I think we also have Japan patent. We can also print protein gradient or growth factor gradient on scaffold that helps in the growth of cells for stem cell growth, we can say. So the concept is that if you have any damaged part, as example, you have some damaged neural cell or spine cell, so you put the stem cell over there and you expect that the stem cell will cover up all the damaged region. But the point is that, or challenge, how it will understand whether it has to go up or go down. So for that, we put some protein gradient or growth factor gradient, and we can print that gradient using printer. So the concept is that if we have a gradient, then from low concentration to high concentration, the stem cell will sense and it move forward. So this is the two ink system to print gradients, and that also works. Okay, now the next challenge, whether we can develop any diagnostic kit for something more meaningful. One example was paper diagnostic for instantaneous blood typing. We know blood typing can be required at any stage of our life. If there is any road accident, if there is any war going on, if there is any pregnancy issues, so that sort of stuff. So the available blood typing machines that we have in the pathological labs require gel column, fiber optic microfluidic device, spin tube method, and TLC immunostaining. And normally in pathological lab, it takes few dollars. However, you have to go to the pathological lab to do that kind of test. So our aim was to develop on-the-spot test, which will give you reliable and accessible results, and that will be also low cost. So we observe that the agglutinated blood transport differently in a porous medium, as example paper. So if we have red blood cell agglutination triggered by antibody-antigen interaction, that decreases blood wicking on paper, and agglutination process enhances chromatographic separation on red blood cell and blood serum. So on basis of that concept, we developed one 3-arm prototype where we treated the blue arm with antibody A, yellow arm with antibody B, and white arm with antibody D. And when we place a drop of blood, then the antigen in blood sample, it interacts with specific antibodies and make agglutination. And if there is any agglutination, then there will be a separation in between red blood cell and serum. So you can see that there are certain level of separations. There are certain level of separations uh, on paper strip A and paper strip D. So that's why we can say it's the A positive blood. And we tried different other technique, like we allow the blood sample reaction with antibodies in a test tube, and then we place that combined solution on paper strip, and we see that the agglutinated blood samples doesn't wick at all. It stays, only the blood serum keep wicking. So we can say, since there is a separation on antibody B and antibody D coated paper, it's a B positive, and the other one is O negative. Then we wanted to make it even more complex or more interactive. Like, can we print, can the paper itself report, rather than having any further interpretation? Can the paper itself report whether it's A positive, B positive, or O positive blood? Okay, and the whole concept came from a very famous movie, Harry Potter. In one of the Harry Potter movies, there was a book. You have to question to the book, and book used to report or answer the question by writing. So we printed antibodies on paper. Remember, in the last concept, we showed how we can print proteins. So we printed antibodies on paper and then allow blood to interact with those printed antibodies so that we can get the reporting in writing. And we also patented that concept. So you see, we have European patent, Chinese patent, and US patent for that. And that is one commercial application, like one pharmaceutical company, they purchase the patent right of our blood diagnostic concept. So I show you a small video how they do it. The company name is Hemokinetics, and their product name is GILF. So this is the product. one has to place a drop of blood in the reaction zone, and the antibody is already printed underneath the reaction zone. So we allow 60 seconds so that the blood antigen can interact with printed antibodies in the reaction zone. And once the interaction is over, like we tentatively give 6 seconds, then we wash the reaction zone with buffer to clean out or wash away any further red blood cell from that. And you see, the device is showing in text what is the blood group. So it's easy, it's low cost, it's accessible. Okay, now the next challenge, can we build up paper diagnostic for pathogen detection as bacteria or virus? The virus is the most difficult part because of the size, and there are not too many viruses that you can work with. If you can remember, the COVID situation happened from one level 3 or level 4 lab of Wuhan. So you cannot work with many of the viruses. So there are only few viruses which you can work with, and which are not very harmful, which are not harmful to human body. So our target was to prepare antibody active paper diagnostics to detect and quantify the presence of viruses, because there can be lots of biological war going on, like SARS virus in 2004. In North America, lots of SARS attack happened through envelopes, like through the mail service. Okay, so if they can detect it beforehand, that'd be really useful. So we wanted to have a detection and quantification technique for viruses. For that, we developed modified sandwich ELISA on paper surface. If you know ELISA, is the enzyme linked immunosorbent assay. So it has enzyme. So in presence of target antigen, for our purpose, it is the virus. It gives color indication. In absence of virus, it will not give any color indication. So, and our technique can detect virus concentration from 10 to the power 0 to 10 to the power 9. So it's a very wide concentration range. And so it was like, since it was virus, it was clumsy. It took really good amount of time. We started with M13 virus. After 6 months of experimentation, all the M13 virus got died away for some sort of infection, and so it took another 6 to 12 months to regrow M13. So during that time, we started working with T7 bacteriophage virus. So the results you are seeing, those are from T7 bacteriophage virus. However, all the results are being published, so you can see the detail, or we can discuss about detail later on. So now, can we also develop paper diagnostic to detect uric acid in urine? You know, uric acid is one of the biomarkers to pre-screen any kidney problem. So the available pathological detection techniques to detect uric acid from urine are mainly spectrophotometric based. If you see on the screen, there are available techniques. Most of them are spectrophotometric based, but then again, you have to send your urine sample to pathological lab, or you have to go to the pathological lab in person. So we studied the whole chemistry. If you see, the whole detection chemistry is there. We studied the whole chemistry. Then we place the chemistry on top of paper. So we build up paper diagnostic by placing the chemistry on paper, and then in presence of uric acid, the paper will show you color. It's a sky blue color. So from color gradient analysis, we can understand what is the concentration of the uric acid. And also it got some sort of media attention, and in national stage, it was a really big hype. So, and also my student who was working on this project, Najib, he is now doing his postdoc in UC Berkeley. And so he tried to develop a computer and cell phone app so that the color analysis can be done using that app. And he was successful to develop that sort of app. And the whole the experimental and the digitalization process we have patented from Bangladesh. So you see, like we always try to have the applied part of the scientific research or engineering research. So next, paper diagnostic to detect formalin in food. So in Bangladesh, a few years back, there were a report that the vendors or the sellers, they're randomly using formalin to preserve food like fish, meat, and dairy products. But constantly exposure or consumption of formalin can lead to respiratory problems and possibly to cancer. So it became a big crisis in the country. So we try to detect formalin or pre-detect formalin using paper diagnostic. You know, formalin is a formaldehyde water solution, 40% volume basis and 37% mass basis. So then again, we studied the chemistry thoroughly. We place the chemistry on paper. So rather than doing it on in a beaker and jar, we did the chemistry on paper. And from the paper diagnostic, we can detect formalin concentration from 0 ppm to 1000 ppm, considering the color gradient, just like a pH paper. Okay. And we tested specificity test, because sometimes formic acid, acetone, glucose, they also give false positive results instead of having formalin. So our specificity test passed the positive detection. And also we tested our results with respect to spectrophotometric technique and also available literature from different sources. And we proposed different prototypes to detect formalin in food or any food solution, and we patented that whole technology. And one pharmaceutical company, they procured the patent right from us, and they tried to do the commercialization. I think they're like, it is still in their pipeline since it is a commercialization part. And during this time frame, government successfully controlled the import of formalin. So they're waiting that if there is any surge in future, they will make the commercialization of that kind of device. We also develop antibacterial paper for health and food applications. We used benzyl conium chloride to develop antibacterial paper, which can be used for health and food applications, especially for food packaging. We can use, and these antibacterial paper can be useful against gram-positive and gram-negative bacteria. We did laboratory trials and a series of experimentation to understand which is the optimum concentration for benzyl conium chloride. And like for both gram-positive bacteria and gram-negative bacteria, we identified the optimum concentration. So I'm not going into the very detail of that, but whoever works on biotechnology, you understand that it requires lots of culturing and cross verification, and like that's all. Okay, the next question. So we studied whether we can print and whether the printed biomolecules are still alive, then whether we can detect whether they are still functional. And the next one, can we do molecular simulation to understand or predict the experimental outcome before we enter into the experimentation? So for that, we handpicked one molecule named picloram. It is a herbicide and picloram antibody. So this is a very basic structure of antibody. If you are familiar with biochemical engineering, you already know the structure of antibody. But for those who are not familiar, I'm just showing once again. So in antibody, you have light chains of amino acids and heavy chain of amino acids. In the light chain, you have 200 to 210 amino acids, and you have constant region and variable region. So in this region, that is called antigen binding fragments, FAB. And within antigen binding fragment, you also have the gray zone, that is the variable region, that is called single chain fragment variable. And in single chain fragment variable, you have different possible interaction zones, which are called complementary determining regions. So basically, in the whole antibody, the CDR, or the single chain fragment variable, is the most important part, because that goes for the antigen antibody interactions. So for picloram and anti-picloram system, from experimental study, we know the amino acid sequence of anti-picloram CDRs. So what we did, we run homology modeling. That is, we took different fragments of amino acid chains, and we use a database, existing structural model, where the fragment matches the best fitted fragment. Okay, like I'm trying to explain how to make it very generalized. So like for each fragment, if we find the best match, then just like Lego playing, just like Lego, we combine all the small fragments together, and then we run force field theory to build up the amino acid fragment, or sorry, the antibody fragment. So this is the force field optimization of our anti-picloram CDR. And if we remove all the molecules, then we see the ribbon-like structure. And in the ribbon-like structure, we find possible binding regions. So it is basically 3D pockets. So in the 3D pockets, we place the picloram and we perform hundreds of permutation combination to understand from hydrophobic point of view, from polar point of view, hydrogen bonding, positive, negative point of view, which one is the best fit. Once we find the best fit, then to get the more accurate information, we run quantum mechanics. Because if we run quantum mechanics, that will give you this van der Waals force surface, or electrostatic potential surface, and also the interaction energies. So through this process, we can run the simulation at pH 7, high pH and low pH, and we can find the binding energy for different combination. And then we go to the experimental part and cross verification whether the simulation results are okay. My headphone is giving me some problem. Is it okay if I change my headphone? Can you please give me 30 seconds? It's saying that the battery is low.
Yes, sure.
Okay, can you hear me now? Can you hear me?
Don't hear your voice yet.
Can you hear me now?
Yes, but the voice is low. Maybe you can.
I can improve.
We
I
can
can
hear
come
you.
close to the
That's good.
screen.
Thank you.
Wonderful. So whatever simulation that we do, that is only to help our experimental studies. So after that, we need to cross verification the results through experimentation. Now, at present, on the biotechnology part, I'm working on different projects like paper-based diagnostics to detect anemia. So you know, anemia is the biomarker to detect different health condition, including cancers. And then micropad, that is micropaper based analytical device for pre-screening of thalassemia, and point of care diagnostic devices for non-alcoholic fatty liver disease, NAFDL. So those are the ongoing projects. I have my master's students working on those projects. So feel free if you ever find those interesting and to have any future collaboration on those projects. Okay, and some just honorary mention, those are not very much applied. Those are more about fundamental research. So we publish, as an editor, I published 2 books in last few years on food, and also run or conducted several investigative research, as example, analysis of pesticide residue in locally grown cauliflower, cabbage, and eggplant, then artificial ripening on banana samples, analyzing ripening agents and change in nutritional parameters, and dental erosion potential of beverage and bottled drinking water in Bangladesh. So those are kind of investigating reports. Those are very good for to understand the current situation of the food safety and security of the country, and it can be replicated in any country. And also a series of other publications in food. So those are, I'm not going into detail, I just mentioned that. One interesting thing I would like to mention, that is we worked on biofuel. So 2 types. One is biodiesel or bioethanol. So we used, we studied growth kinetics of high lipid content algae in local environment. We tried to grow a very high lipid content algae under different conditions and different growth media. And also we produces a very high quality bioethanol from maize that was 99.5% ethanol, and we studied different fuel properties of that bioethanol so that it can be used as the octane booster and can be mixed with octane and diesel in future. Now, industrial sustainability. So that is the next part of my presentation, and I'm quite thrilled to give this presentation because there is huge opportunity for chemical engineers to work in industrial sustainability. And since I have been working with industries for more than 10 years, we understand that the future, if you see from this graph there, the green technology and sustainability market global forecast to 2030, it's increasing. So it will exponentially increase in the upcoming years because European countries, USA, and many other countries, they're in a process to reduce their greenhouse gas emission through the production process or from the production process. So they have to make sure their technology is green, and they have to make sure they use low resources, they produce low greenhouse gas emission, they produce low waste or they use sustainable raw materials. So industrial sustainability is also known as sustainable industries or industrial sustainable production. The phrase refers to greening of energy intensive industries like steel mills, pharmaceuticals, textile, petroleum. So I'm pretty much sure you have in Iran, you have all those kind of industries. And this research or study is associated with water footprint, carbon footprint, and ecological footprint. And to understand the industrial sustainability, there are some toolkits which are energy efficiency, greenhouse gas emission, water, carbon, and ecological footprint, and life cycle analysis. So industrial sustainability research, it is kind of a 3-dimensional research approach. So to understand that kind of research, we have to understand the industrial process from inside, very in-depth understanding of industrial process and different aspects of industrial process. So if you see, we run a series of research. One research was evaluation of present and future wastewater impacts of textile dyeing industries in Bangladesh. So to start with, I started with textile and leather because in Bangladesh, in textile, Bangladesh is the second largest apparel manufacturing countries in the world. So in apparel, we're a big fish. And every year we export about $50 billion, okay, on an average. So there are lots of working opportunities there. And then leather is another potential item where we can contribute, and that can be another export item for our country. So we also studied technical and economic aspects of reusing textile effluents as processed water. So everything is connected. Identifying practices and prospects of chemical safety and security in Bangladesh textile sectors, and a material balance approach for assessing textile chemical consumption, a case study of Bangladesh textile sector. So those studies are conducted just to understand the sector from inside out. And in this study, you can see that we studied the environmental impact through the wastewater contributed by textile dyeing sector. So we conducted that study for 10 years, 2011 to 2021. So it can be extended for another 10 years. And then the major part, we studied water footprint management for sustainable growth of the apparel sector and also the leather sector. So now most of us, we know about carbon footprint. Some of us, we also know about water footprint, and there is another footprint called ecological footprint. So we started with water footprint. In water footprint, you have three components. One is blue water footprint, that is the surface or groundwater consumption. Another is green water footprint, that is the rainwater consumption. And another is gray water footprint, that is the wastage, the wastewater, and whenever the wastewater mixed with any fresh water, it pollutes that fresh water as well. So the whole combination is considered as gray water footprint. So the analyzing of total water footprint is simple. We calculate blue water, green water, and gray water footprint for an industrial process, and then we divide by the number of unit produced against that water footprint, so that we can identify water footprint for each unit. Now, as an example, to calculate water footprint for apparel sector, we need to study the manufacturing process of textile. You see that it starts with natural fiber or regenerated fiber, crop or polymerization, then fiber preparation. Here we have dyeing process, spinning, yarn manufacturing, weaving, fabric finishing, and garment manufacturing. Each stage we have different kinds of dyeing. So to calculate water footprint for textile, we need to calculate water footprint for cotton cultivation, transportation, wet processing, and water footprint contributed by workers, because it's very much worker intensive industry. So to calculate water footprint for wet processing for apparel sector or textile, there are different wet processing, one for knit dyeing, another oven dyeing, another denim dyeing, and then denim wash. In knit dyeing, we also have colored fabric, white fabric, and polyester. So you have to understand the process sequence and what are the chemicals used at different stage of the process sequence. So we had to stay inside of the factory and multiple factories to study. One of my PhD students is working on textile process, and hopefully he will submit his PhD thesis by early of next year. And then process sequence of cotton oven dyeing. So it's an oven dyeing. So we also need to understand the oven dyeing sequence, and we also need to understand the denim dyeing sequence, whether it's for indigo dyeing or it is for rope dyeing. Once we understand the whole process, then we try to calculate what are the amount of fabric we produced inside our country and amount of fabric we imported from other countries. So you see those numbers, and then we have to calculate water footprint for cotton cultivation. Then again, cotton cultivation can be done for our imported cotton and cotton cultivated inside our country. So we have to calculate blue water footprint for cotton cultivation, green water footprint, and gray water footprint. And this is the internal water footprint for cotton and external water footprint for cotton. So internal means the cotton produced inside the country. External means cotton imported from other countries. So we have to also count those water footprints. And then for wet processing, we can calculate the blue water footprint for textile industries, gray water footprint, and total water footprint. For total water, we can also consider internal, that is fabric produced inside of the country, and then external, which is fabric imported from other countries. And once we know all those numbers, all the data from all sectors and all production process, then we can see that the major contributor of water footprint in textile is cotton cultivation. So if we can reduce a significant part of cotton cultivation or water requirement of cotton cultivation, that will obviously reduce our water footprint. That's why man-made fiber is becoming in very high demand in these days, because that requires less water, synthetic fiber. So another example is leather processing. If you see the detailing of leather processing, it requires beam house operation, tan yard operation, post tanning operation, and finishing. In beam house operation, you have soaking, liming, fleshing. Tan yard operation, you have deliming, pickling, degreasing, tanning. In post tanning operation, you have samming, shaving, wet black, re-chroming. So you have all detailed stages. And to calculate water footprint for leather, we need to understand water footprint for feed crop, water footprint of hides and skins, water footprint for beam house operations, tan yard operations, and post harvest operation. And one major problem working with leather, it's a data is not very much available, and leather industries, they're very shy to give data. Okay, so that was the biggest challenge that we had, but we built up good relationship with local industries. We got access, we collected different wastewater and different samples, and we analyzed in our lab so that we can use those numbers in our water footprint calculation. So similarly, we calculated gray water footprint and blue water footprint. It doesn't require much of the green water footprint. So we combine gray and blue, and then we get the total water footprint. And from data analysis, we can understand which process contributes what amount of water footprint. However, once we combine all the numbers, then we find that again the farming sector, that contributes about 92% of the whole water footprint. That means if we can reduce water footprint from the farming sector, the sector will be more sustainable. Now, as I mentioned, the industrial sustainability sector seems to be very productive, and we secured a good amount of research grant from different grant agencies. Like from European grants, I have secured about $1.2 million for WHO, and few other small grants I secured about $300,000. So, and then I build up the Textile Analytical Laboratory. Though the name is Textile Analytical Laboratory, however, we can run few of the food-related research in that lab as well. My ongoing projects are ecological footprint of textile, leather, and pharmaceutical sectors, comprehensive assessment of greenhouse gas emission of apparel sector, and development of medical textiles from environment-friendly natural polymers incorporating with nanotechnology. So in this sector, we are using hemp because hemp is carbon neutral. That's why I place that in the environmental part. And I recently created another research group that is called ISR. So it is the meaning of Industrial Sustainability Research Group. So now I run basically two research groups for the bio and environment. I have BET group, and for sustainability I have ISR, because my understanding, this is the future, and the chemical engineers can make a big contribution there. They understand the process inside out. They understand industrial process better than anyone else. So in future research project, I keep working on point of care diagnostic, medical textile, and biodegradable polymer and biofuel. And for industrial sustainability, I have ecological footprint, greenhouse gas emission, and sustainable raw materials. Right now, I have 4 PhD students, couple of master students, and since BUET, is very much undergrad oriented university. So I have quite a few very energetic undergrad research students. I would like to continue industry academia collaboration in textile, pharmaceuticals, and biomedical manufacturing. Possible collaboration with Canada, McMaster and McGill. Australia, we have Monash University. USA is TAMU. Denmark is GTU. Hungary is University of Győr. In fact, they have established their first Institute of Industrial Sustainability, and they elected me as a board member. I'm going there to give a keynote lecture in early of November in University of Győr. So I'm expecting very high from Sharif University to have future research collaboration. I have, through the industries, I have good contact with China, CNBG, and Oman, Sultan Qaboos University. And if you see, I strategically identify point of care diagnostic because the industrial application is increasing every year. Then global biofuel market, the industrial application is increasing every year. And again, green technology, you see, it is just jumping every year because as per Paris Agreement, every year an industry requires to reduce their carbon emission up to 4 to 6%, so that by 2030 and 2035, they can reduce global warming by 1.5 degrees Celsius. So every industry today or tomorrow, they have to reduce their consumption and greenhouse gas emission. So that's pretty much all. I'm very sorry for the technical glitch and sound related issues. I like to acknowledge my research groups at Buet, McGill, and Monash, research collaborators that we already have, research funding agencies who just heavily contributed in my research, industrial partners. Industries are very shy. They don't want to share any internal information, but I'm lucky enough to work with industries for last 10 years and earn their faith, so we can get all the industry ready data, but we always respect their confidentiality so that we never mention the name of the industry. We always say industry A, industry B, and whenever possible, if there is any opportunity to improve their process, we try to help them. And from Sharif University of Technology, I would like to thank Dr. Hania Safari for continuous communication and collaboration, and also Mr. Yasir Khanjani for inviting me and to build up all those networks, and also School of Chemical and Petroleum Engineering for giving me this wonderful opportunity. We have very high respect about SUT in Bangladesh, and also in my mind, I had friends from Sharif University of Technology, and they're doing wonderfully well in overseas. So we have very good faith and respect of your potential. Thank you very much for inviting me. And for any kind of question, future collaboration, I'm happy to answer. Thank you.
Thank you so much for the talk. We enjoyed it pretty much. And thank you again for accepting our invitation at SUT. I think first we will take questions from the audience that are present in the conference hall, and then online audience and folks can type their questions in the chat box if there are any. Guys, do you have any questions or anything? Okay, seems like we don't have any questions from the in-person audiences. If there is anyone who has a question, in the online system, they can raise their hand and we will give them access to ask their question via microphone. Okay, looks like there isn't any questions. All right.
Was it too big or too boring
There are not
for the audience?
Any questions? I guess we can close the session. We hope to have you here in person, Dr. Mohidos, hopefully sometime soon, to start up the collaborations, and we will keep in touch to, kickstart the collaborations between our university and Bangladesh. We are pretty excited for that.
Absolutely, and it's the same kind of feelings from my end. We would like to invite you to visit Bangladesh. Right now, we're having some political issues, but I hope it will be resolved in the upcoming months. And then both parties from SUT and BUET, we can have very strong collaboration, and we can invite your faculty members and students in Bangladesh.
Yep, we are going to be pretty excited for that. And we hope that the issues in Bangladesh get solved pretty soon so we can have our collaboration started. Thank you again for being with us today. We enjoyed it.
Thank you for the invitation. Thanks a lot. Bye bye.
Thank you so much. Thank you.